So yeah, thank you very much, Julio, for inviting me and for setting up this beautiful meeting. I'm the one between you and lunch. I'm sorry for that. From my experience with the last IRIS meeting, I suspected that I'm supposed to give you a broad overview of what we are interested in currently. And so I'm going to tell you more from the helicopter perspective what these wonderful people are currently working on. And with the idea that we hopefully find possibilities to collaborate in the future. So I'm a bit proud that we were able to establish over the past years the complete translational pipeline from studies on molecular interactions over primary human and cell and murine cells, organoids, organ-on-chip models, and human lung slices that we get from the OR over in silico models, isolated, perfused, and ventilated mouse lungs, and different kinds of pneumonia models in mice, as well as 2 different COVID models in hamsters, to then interventional trials, and backwards also from observational trials and cohorts and patient samples, then back to molecular interactions. So my agenda is to briefly talk about some of the cohorts we have and see whether maybe you would be interested in using data and samples from these cohorts, which I would appreciate very much. Then briefly highlight a few of the basic science projects and then give one example of the back and forth translation. I will start with the cohorts, and maybe you are aware of CAPNETS, which has been running now for around about 20 years. We included around about 15,000 patients. And this started off as a BMB funded by the German Ministry of Education and Research, is nowadays a foundation. And we are always interested in providing data and samples. So if you're interested, just let us know. Same holds true for PROGRESS, which has the aim to define biomarkers for progression to lung injury. Here we have more than 2,000 patients included with longitudinal sampling, doing a lot of omics analysis, and all these data are free for the research community. When COVID came around the corner, we, on the basis of PROGRESS and CAPNETS, started PROVID, more or less the same, but for COVID. And this was the lucky times when the federal ministers came to your lab to give you the check because they were so happy that someone did corona virus research. And in this, we sampled 600 patients or a bit more, and had the aim to evaluate the disease course to understand why endothelial barrier dysfunction happens, to do some molecular pathology and viral genome analysis. And we then used this as a blueprint for a by far larger cohort, and this is the network University Medicine-based National Pandemia Cohort Network, which consists of 3 different cohorts. One is a high-resolution cohort with a very high granularity. One is a sector overarching cohort, not only sampling in university clinics, but also in smaller clinics and outpatient clinics. And one is the population-based cohort. In NAPCON, all German university clinics contribute. And so we now have like 7,000 samples and supplied around about 120 projects with patient data and biosamples, and hopefully many more to come. And we were able to get some additional funding for a large-scale omics analysis from around about 800 patients with longitudinal samples, and to do genomics and transcriptomics, proteomics, metabolomics, epigenomics, and so on. And again, all these data will be available around about July or August this year, and we are happy to provide them to anyone who is interested. Something completely different we did in the past few years is to establish the single lung cell transcriptomics pipeline and to analyze cross-species. So to compare the hamster model or different hamster models with the human single cell transcriptomics, and with this to get the possibility to really deep dive into pathomechanisms. And to see, when you find something in the hamsters, is this really important for human beings as well? You cannot get longitudinal sampling of lungs from patients, but if you have the hamsters and you have the longitudinal samples from the blood from the patients, you can see whether a finding from the hamsters is also important for human beings or not. We also stepped into systems medicine of community-acquired pneumonia with the project CAPSIS, again funded by the German Ministry of Education and Research. And here we use large patient data banks on the one hand, and mirroring the course of the disease in different kinds of animal models, and bring these data together in mathematical models to hopefully get a better insight into what is happening between pneumonia and acute lung injury. And we used this as a blueprint for SYMPATH, which is systems medicine of pneumonia-aggravated atherosclerosis. And so I was very happy to hear the talk by Rosario earlier today, because we are very interested in the pathomechanisms between pneumonia and atherosclerosis or cardiovascular events then occurring. Again, we use our patient cohorts from pneumonia patients as well as from cardiovascular patients. And we use different kinds of animal models that are prone to atherosclerosis, and then bring this together in mathematical models and hopefully come up with more interesting therapies than, let's say, aspirin. Something completely different is a project that aims at establishing phage therapy for Pseudomonas in Germany. If you want to establish that, you need a real medication, a drug that can get approval. And so our pipeline starts with wastewater sampling, and the colleagues from the DSMZ in Braunschweig, they love wastewater from clinics because there they find bacteriophages against multi-resistant bacteria, and they isolate them. We now use a cocktail of 3 phages. These 3 are genome sequenced and produced under P conditions. And we did the preclinical evaluation and are currently waiting for the approval for the phase 1 trial, and hopefully go into phase 2, a very small phase 2, because we do not have much money, in the beginning of next year. The aim is to treat patients with bronchiectasis, CF and non-CF bronchiectasis, and a constant colonization with Pseudomonas aeruginosa in order to reduce the bacterial load. So relatively low-hanging fruits. I'm pretty optimistic, but I'm aware of many hurdles that we are facing, like mucus, like biofilm, and all that stuff. So yeah, maybe a few words to basic science projects. Something that really drives me is the question, why do get patients that are mechanically ventilated Pseudomonas infections and I do not? We elaborated on that when Tony gave his talk. And one possibility could be that the glycocalyx is different under mechanical ventilation as compared to a normal breathing. And I'm very happy to have a project together with Peter Seeberger, who is an extremely skilled chemist running a large chemistry department. He is an expert of sugars. And so we teamed up and are trying to understand what is happening under different conditions with the glycocalyx. What about the pathogen glycocalyx interaction, the role of fucosylation, and so on. And in order to study that also in an animal model, Matthias Felten established a model of ventilator-associated pneumonia. And what is special about this model is that he really ventilates the mice and then weans them from the ventilator so they spontaneously breathe afterwards. And we can see what is the impact of the mechanical ventilation on the course of pneumonia afterwards. And what he sees is that mechanical ventilation over only 4 hours in these mice clearly increases the susceptibility to Pseudomonas aeruginosa, and that this goes along with an acidification in the alveolar compartment. And this acidification is due to a blocking of the sodium-hydrogen exchanger. And so we here see a target that we can indeed use, hopefully, to prevent from this alveolar acidification and thereby from the increased susceptibility to Pseudomonas. We did quite a number of in vivo and in vitro studies on that. I obviously cannot show all data now. A completely different story is on CFTR. You know CFTR from the epithelial cells, but it also plays a major role in the endothelial cells. We have shown that for smooth muscle cells in the vascular department as well, in some earlier years. And here we see that in pneumonia, there is a loss of endothelial CFTR that drives barrier failure and edema formation. And if we give a CFTR potentiator, we can reduce dramatically the barrier failure and the edema formation. And as you know, there are potentiators out on the market, and we are now trying to find a way to do a clinical trial with CFTR potentiators. And finally, what is extremely interesting from my point of view is that there is an important impact of the circadian rhythm on the lung injury under mechanical ventilation. As you all know, in most ICUs, you have light at daytime and also at nighttime. There is noise at nighttime. Feeding is 24/7. So there is a clock disruption in most patients, and this indeed drives ventilator-induced lung injury. There are specific molecular targets, few of them we were able to identify, and we think that this is indeed a possibility to reduce lung injury under mechanical ventilation. So I showed you this translational pipeline, and in the last minute, I would like to give you one example how it can work. And this was when COVID came around the corner and we had the first 40 patients treated in our department. We used the samples, the blood samples of these patients for ultra-high throughput clinical proteomics, which means that the colleagues from the biochemistry department with this method can do a 500 protein analysis overnight. And what they showed was, well, there was a lot of inflammation and coagulation, but there was a lot of complement activation as well. And this intrigued us because we at the same time just published a paper showing that C5a neutralization can protect, in this case, mice with pneumococcal pulmonary sepsis. So these were really severely, mice with really severe pneumonia that needed to go on our mouse ICU and needed to be mechanically ventilated. And these were effectively treated with a C5a Spiegelmüller in this case. There was a lot of other evidence that C5 could be an important target. One is a study that was done by Birgit Sawicki from our immunology department, who showed that there is a connection between the complement cascade T cells and endothelial inflammation. And so we spoke with a company that produces a C5a antibody, which is called Inflarix. And we tried to set up a clinical trial in severe COVID-19 in Berlin. Unfortunately, the ethics committee didn't like the idea. So we had to move to the Netherlands, where it took us 5 days to set up a trial. And Alexander Vla and his colleagues did a great job in performing this phase 2 trial, which showed safety and some hints of efficacy. And then there was the phase 3 trial that was then published last year, which clearly showed a reduction of all-cause mortality in patients with ARDS due to COVID-19. So with this, I'm at the end. I would again like to stress, if you're interested in data or biosamples, just let me know, and thank you for your attention.
Very good, excellent. So many topics in 10 minutes. This is we have a full conference. A lot of interesting things. Okay.
So amazing scope of work, Martin. 2 questions, I guess. I've lost track of how many editorials I've written on drugs that looked good in animals and then completely failed in humans. And we heard in Tony Torres's talk, they've gone to some lengths to try and get away from the single massive inoculum. And it seems to me that there's a fundamental problem with animal models that's not really pneumonia as humans understand it, and your observations on that, and whether there are new tools or we're just stuck with bad tools. The second question relates to actually, no, I'll just leave it with that. That's
complex enough. Thanks. So I'm not sure whether I completely answer your question, but what I think is, first of all, we should not rely on one animal model. We need more different kinds of models. And so, for example, if we're talking about ventilator-induced lung injury, we also use human cells that are chronically, periodically stretched. Or if we're talking about a endothelial barrier stabilizing effect in a mouse model, I would always argue you need a prophylactic approach, but you also need a therapeutic approach in a mouse with pneumonia, but not only pneumococcal, maybe also influenza, maybe something else. You need also the model of pneumonia plus mechanical ventilation, because that's the situation your patient will be in, maybe plus antibiotic treatment. And if this, in all these models, you see a positive effect, then I get more and more confident that it might make sense to go into the patient as well. This does not answer your question regarding the inoculum. I'm not completely sure whether it is so important, whether there is a larger or smaller inoculum that induces a pneumonia that is an alike of what you see in the human patient. I mean, in the end, the question is this, when we're talking about pneumococcal pneumonia, a patchy pneumonia with bacteremia and in the end sepsis, as we see it in untreated human beings? I mean, if I succeed in getting this clinical picture in the mouse, is it really so important whether this, is
so I guess I disagree slightly in that I'd say this is a little bit like idiopathic pulmonary fibrosis, where they use bleomycin models. It produces the same pathology, but it's not the same disease. And particularly if we're looking at the immune response and how there's clearly in a human, there's this adaptive immune response, which is very different to an acute lung injury. And so I'm not saying they're invalid models. I'm just saying I think there's a fundamental limitation that is stopping us from making better decisions about how many drugs we take from a animal to a human, and are we missing potential targets because we've just overwhelmed the human response, and we don't actually have we're not understanding the therapeutic window for some of the immunology that's happening.
Yeah, we do have the double hit model established of influenza infection and then pneumococcal infection. If you have influenza infection first, you need like just a tiny bit of pneumococcal pneumonia to induce, of pneumococci to induce pneumococcal pneumonia. Nevertheless, if I would have the choice, take this model in addition to normal pneumococcal high load infection, or use two different kinds of pneumonia models, maybe plus mechanical ventilation, I would always go for the latter ones, because we do not see a huge difference in the course of pneumococcal pneumonia, no matter whether they have been infected with a high bacterial load without influenza or with a low bacterial load with preceding influenza infection. So I fully agree. You should not rely on one model with a high bacterial load, but maybe this is not the most important difference from my point of view.
Yes. Oh, I'm sorry.
Okay, thanks, Martin. Great talk, like always. I have two concrete questions. One is the vilobelimab story is really cool, but a little bit late for the COVID-19. So what are your plans? Which RDS you want to try next? And the second one is CFTR modulation. Should be easy to translate into clinical practice as the compounds are already available. But as I understood, they prevent maybe ARDS in pneumonia. So you want to try it in late pneumonia with ARDS or in moderate pneumonia to prevent ARDS, but then you have a cost issue?
Yes, very good questions, as always. Regarding the first one, as I showed, we have, from my point of view, pretty convincing data that this is not only, that C5a is not only important in COVID-19, but also in, for example, strep pneumonia. There are clinical data out of for sepsis. And so I think the companies Influrix, which is on the C5a antibody, and Aptarion, which is on the C5a Spiegelmeer. They might go into the direction of bacterial or any cause of severe pneumonia. Regarding the second question, this is a cost issue, but it will not always be a cost issue. And so I think it could be wise to use it in severe pneumonia to prevent from going to the ventilator.
Thanks, Martin, for your talk. Just going back to the initial conversation that Grant raised, you know, I think the beauty of your sort of approach, going back from humans to animals and back and forth, probably would have, you know, added layers of validation, which is what was needed. So we currently have a sort of an animal experimental model for bacterial pneumonia, that was approved on bacterial infections. I was probably going to reach out to you to see if you're interested in participating, which is actually addressing this question: what doses, what's the model, what's the right thing for the investigators to do to as much get information from these models to reflect what humans can happening in humans. And so I think going to your point, Grant, you know, how are these, can we all as an investigators come together and come up with some consensus to see how best do the next, you know, bacterial infection models to represent humans. And so I think it's needed in the field. I think all
a consensus statement of the ERS on, you know, what kind of preclinical models should you use. And I think everyone in the room agreed on that. It's not the one model, it's the number of models that you somehow smartly bring together that gives you confidence in your drug.
Very good. Yes.
To know whether there is any synergistic effect between anti-C5a and other anti-drug inhibitors in the matter of community-acquired pneumonia. Yes, and the reason why I'm mentioning that was as in PET, we could get an inhibitor of other two receptors for interleukin 8, which we are thinking right now for community-acquired pneumonia and ARDS. Just as in theory, as a concept, I'm wondering, combining them together, would it have a synergistic effect against this product?
Yeah.
So, something to think about.
Yes. My personal point of view is it must make sense. It is obvious that it must make sense to address the inflammatory root on the one hand and the endothelial barrier dysfunction on the other hand, and maybe something like complement coagulation and so on in the more severe patients as a third pillar. I think the challenge is to find the right patients. So which patient may benefit from anti-inflammation, which one from anti-endothelial barrier dysfunction, yeah, not to do harm to anyone in this scenario.
Very good, very good, very good comments. Yes?
Just to tease you out on bacteriophages, it seems like everybody wants to start a bacteriophage lab and lots of companies. The problem is that the data I've seen shows that, you know, bacteria and viruses have been fighting a war a lot longer than humans have been around, and bacteria develop resistance to bacteriophages quite quickly. In fact, I know 2 companies who promote that as an advantage because you'll have to keep buying their product every 3 or 4 weeks. So I can see in an acute infection how bacteriophages may be very useful, but I have some skepticism about their real utility in chronic Pseudomonas infection like CF and bronchiectasis. Do you have any thoughts or comments on where we should take it?
Wonderful comment. You elucidated every problem that we have in the field. So yes, resistance is a major problem, and there are many, many others with phages. It's interesting to see that after 100 years of phage use in Eastern Europe, within the past 5 years, everywhere someone popped out and said, wow, I'm the phage expert. We have been starting in 2003 or so with endolysins, so the phage license that can kill bacteria. You may be aware of the clinical trial by, don't remember the name, in a US company that was very positive in phase 2 against Staph aureus, right-sided endocarditis and bacteremia, and in the phase 3 they failed. And I think it's extremely challenging to be successful with phages or phage license. And that's why I said we are aware of many, many hurdles that we have to overcome. It's resistance, it's getting the phage to the right place. And I'm absolutely not sure whether this in the end works. What worries me is that there are so many people now starting to treat with phages that come from sources where they do not know at all what they have in their vial. There is a lot of LPS in there, that's for sure. There is no evidence for efficacy or safety. So I think we have to go this way, but I would not give my own money in.
And you obtain the phages from the sewer that is coming from the hospital.
Yes, they find the phages in the wastewater of hospitals mostly.
Yeah.
Then they characterize them. We used like 400 different Pseudomonas strains and I don't know how many phages, and looked for a cocktail of 3. It should not be too many. Then they are produced under GMP conditions to make sure that this is a really safe or a really
But you're assuming,
product.
because I was trying to figure out from where to get the virus that is going to kill the Pseudomonas. But I was thinking that in the hospital, Pseudomonas are so predominant. I was thinking that these Pseudomonas surviving, this is the one that killed everything. But you see, I was trying to, from where I get the.
No, I mean, if there is no Pseudomonas around, you will not find a phage
Of course,
against
yes.
Pseudomonas. So
Then essentially you have to.
Pseudomonas
How do you know that these phage that then you test all the phages to see who is killing the Pseudomonas?
Yes.
Okay. Yeah, you need to see. Yes.
Yes, that's one of, maybe not everyone could hear it. The question is, phages can make a bacteria more susceptible to antibiotics, and that's a very, very valid point. Nevertheless, this is in vitro findings. We do not know whether this is important in patients, like the observation that phages can invade biofilms. Yes, they do, but it's still by far more difficult for them to reach the bacterium if there are biofilms. So, yeah. Yes, there are case reports out, but no clinical trials that are controlled. Yes.
Very good. Well, thank you very much, Martin.
you.
As always, very good.